Imagine, for just a second, that you were tasked with drawing a perfectly accurate map of a major city's entire traffic grid.
Oh, wow.
Yeah, so every stoplight, every one-way street, every hidden alleyway. But, and this is a massive but, there is a catch.
There always is.
Right. You are not allowed to look at a map, and you cannot, use a drone or a helicopter to just look down at it.
Okay, so how do you do it?
Well, the only way you can figure out how the traffic flows is by randomly dropping these massive concrete barricades into different intersections. You completely close them off, and then you just sort of sit back and watch where all the angry, honking cars end up an hour later.
That is a pretty chaotic way to approach urban planning. I mean,
yeah.
You block Main Street and suddenly, a tiny residential road 3 miles away is just completely deadlocked.
Exactly.
And you are left trying to deduce the exact chain of detours that caused that specific bottleneck, which is, practically impossible.
It is practically impossible to get a clear picture from that kind of indirect observation. But wild as it sounds, that is exactly what biologists have been trying to do for decades.
Yeah, they really have.
It's how they've been mapping out the decision-making processes of human cells. And today we are doing a deep dive into a really fascinating 2026 paper. It was published in the journal Cell, led by researchers Julong Jiang, Cece Chen, Matt Thompson, and their whole team.
It's a brilliant piece of work.
It really is. So we are going to explore their new AI and computational framework. It's called DSP. And the mission here, for you listening, is to understand how this tool takes that incredibly chaotic indirect data and, finally lets us draw a high-definition map of cellular behavior.
Yeah, for anyone who is even remotely curious about the future of medicine or biotechnology, or honestly just how complex systems work without just falling apart, this is huge. It really reveals the source code of cellular behavior.
Okay, let's unpack this, because before we get to the solution, D's been pine, we need to understand the problem. Why is mapping a cell's internal logic so notoriously difficult?
Right, so to appreciate what DSPIN achieves, we first need to look at the limitations of how we've been analyzing gene regulatory networks, or GRNs.
GRNs, right.
Yeah. Cells use these networks to basically maintain homeostasis and respond to their environment. And historically, mapping these networks involved relying heavily on single-cell mRNA sequencing data.
Okay.
But data collected across thousands of different perturbations. So you basically knock out a gene, or you alter a signaling condition, or apply a drug, and then you measure the resulting expression levels across the rest of the cell.
Which brings us right back to that traffic analogy. Yeah. You are applying a biochemical barricade and measuring the fallout.
Exactly. And scientists have, they've built plenty of computational methods to parse that fallout. Algorithms like GDA3 or PADC or GRNBoost2.
And those tools are mostly looking for direct links, right?
Yeah, they typically hunt for patterns, like mutual information or direct pairwise correlations between genes to try and reverse engineer the network.
But those traditional algorithms run into a major roadblock, right? Something called the masking effect.
Yes, the masking effect is a massive headache. Because biological pathways, they do not operate in a vacuum. They overlap. They compensate for one another.
Right.
So let's say gene A has this strong activating control over gene B. It turns it on. In a localized, perfectly isolated system, that correlation would be super obvious.
Just a straight line from A to B.
Right. But what if a perturbation induces a much broader cellular stress response? A totally parallel pathway might broadly suppress the entire region where gene B resides.
It's like trying to figure out the recipe for a soup by only tasting the finished bowl while someone keeps throwing in random spices to confuse you.
That is a perfect way to put it. The direct activation signal from gene A to gene B gets completely drowned out by the broader systemic suppression.
Because the algorithm just misses it.
It registers a weak, completely statistically insignificant relationship. It misses a crucial regulatory link entirely. And what's fascinating here is that this masking effect is uniquely devastating when it comes to finding those activating interactions.
Wait, really? Just the activating ones?
Yeah. Traditional algorithms have generally been okay at finding inhibitory interactions like cases where one gene turns another gene off. But they routinely fail to detect the positive activating connections.
Why is there a difference? I mean, a signal is a signal, whether it's turning a pathway on or shutting it down, right?
You would think so, but it comes down to the fundamentally destructive nature of the testing process itself. When you apply a massive perturbation to a cell, You inherently disrupt its overarching modular structure.
Ah, I see.
Yeah, you break the coordinated localized effort of these functional gene clusters. So the localized positive activating signals within those modules, they just scatter into the background noise of the disrupted transcriptome.
So you only see the big negative effects.
Exactly. The only signals strong enough to consistently register above all that noise are the negative braking actions, the inhibitory interactions. You only end up seeing the systems failing rather than the systems building.
Man, so relying on pairwise correlations clearly fails when the biological system gets stressed and that modular structure just shatters. So how do we get around this masting effect? Because the researchers behind DSPine had to abandon that old approach entirely.
They did. They had to look outside of biology, actually.
Yeah.
They turned to statistical physics to find a way out of the noise.
Okay, wait. Quantum spin in human cells? Are we just throwing physics buzzwords at biology now?
I know it sounds like buzzword bingo,
Yeah.
but it's not quantum physics, it's statistical mechanics. They adopted a framework known as a spin network model, specifically something called a Markov random field.
Okay, so how does that work in this context?
Well, in physics, these models were developed to understand complex systems like magnetic materials. Imagine you have millions of atoms, and the magnetic spin state of one atom directly influences the state of its neighbors.
Okay, I'm following.
It is a way to mathematically describe how a massive collection of interacting variables eventually settles into an organized global state.
I mean, I get the magnet analogy in theory. A spin pointing up or down maps neatly to the concept of a gene being turned on or off. But biology is rarely that binary, right?
Well, it's definitely not.
Genes operate on this massive gradient of expression levels. It's not just a simple on or off switch. So how does a framework built for binary magnetic states capture the nuanced, continuous nature of a living cell?
And that, right there, is the major conceptual leap the researchers had to make with D10. It actually stands for Dimension Scalable Single Cell Perturbation Integration Network, by the way.
AVSP? Got it.
Right. So DSPN does not treat gene expression as a binary variable. It adapts the physics framework to use continuous state variables. It basically maps the entire energy landscape of the cell.
Energy landscape, that sounds very sci-fi.
Think of all the cell's possible states as a sprawling topological map, like a physical landscape of hills and valleys. The valleys represent stable states of homeostasis. It's what physicists call basins of attraction.
So the framework treats the underlying gene regulatory network as the fixed topography of that landscape. The hills and the valleys are already there.
Precisely. And they utilize a concept called a hop field network to make sense of it. It's an early neural network model associated with associative memory in the brain.
Wait, associative memory, like when you smell something and it triggers a childhood memory.
Exactly like that. When you experience a specific input like that familiar scent, it excites a specific pattern of neurons. And that causes your brain's network to settle into a pre-existing basin of attraction, which you experience as retrieve memory.
Oh, that's wild.
Yeah. DSPMine models cellular perturbations through the exact same mathematical lens.
Okay, so the perturbations say introducing a toxic drug to the cell that acts as the input, like the scent.
Yes. It selectively excites or represses specific parts of the network, and it forces the cell to explore different transcriptional landscapes, to shift its operations and settle into a new valley, a new state of homeostasis, just to survive the stress.
So DSPI isn't just looking at whether gene A correlates with gene B. It is modeling the underlying physical rules that govern how the entire system rolls down that hill into a new stable state.
You've got it. It integrates the perturbation not as a nuisance that creates noise, but as a defined force pushing the system across the energy landscape.
That is so smart.
It is. By doing this, DSPen separates the localized gene interactions from the global state shift, which effectively strips away the masking effect that completely blinded those older algorithms.
Okay, I love the theory here. But modeling the continuous energy landscape of a network with thousands of genes sounds computationally intractable. I mean, even a massive supercomputer would choke on calculating the permutations of that many continuous interacting variables.
Oh, absolutely.
So they must be cutting corners somewhere to actually make the math feasible.
Right? You've identified the exact bottleneck. This is what has historically prevented statistical physics from being applied to large-scale transcriptomic data.
Because the math is just too heavy.
Way too heavy. Calculating the exact joint probability distribution of the entire network requires computing what is called a partition function.
A partition function.
Yeah, which basically means summing over every possible state of the network. For a genome-scale network with thousands of genes, that is practically mathematically impossible.
So how does DSPIN not just crash immediately?
DSPN bypasses this using a brilliant mathematical trick called pseudo-likelihood.
Pseudo-likelihood. Okay, how does that avoid the impossible math of the partition function?
Well, instead of trying to calculate the probability of the entire global network state all at once, pseudo-likelihood breaks the problem down into manageable chunks.
Like solving a puzzle one piece at a time.
Kind of. It estimates the conditional probability of each individual gene's expression, given the known expression levels of its immediate neighbors in the network.
Oh, okay, so just looking locally.
Right. By focusing on localized conditional dependence, it avoids that intractable global calculation. But it still yields a highly accurate approximation of the overall network structure.
It is a localized approximation that saves immense computational power. I get that. But even with that shortcut, interpreting the resulting model has to be a nightmare.
It is definitely a lot of data.
Right, because staring at an approximated energy landscape of 10,000 distinct genes isn't inherently useful to a human researcher who's just trying to understand a specific disease pathway.
Exactly, which is why DSPN integrates a structural hierarchy. Before mapping the intricate network, it zooms out. It groups individual highly correlated genes into what they call gene programs.
How does it group them?
It uses a technique called orthogonal non-negative matrix factorization, or ONNMF.
Okay, ONMF. We see matrix factorization a lot in machine learning, but does ONMF just clump genes together based on proximity, or does it actively filter the signals?
It actively filters them, which is the beauty of it. By using matrix factorization, ONNMF mathematically untangles overlapping pathways.
Give me an example of how that works in the cell.
Sure.
Yeah.
So if a cell is undergoing cellular division while simultaneously responding to a toxin, the gene expression signals from those two events overlap heavily. It's a mess.
Right.
ONNMF separates those latent variables. It isolates the distinct volume of the division signal from the toxin signal, even when they're happening concurrently.
Wow. So it untangles the mess. And then from there, the paper discusses how DSPIN uses a concept called coarse graining to group those gene programs into massive functional modules.
It's coarse graining.
Here is where it gets really interesting to me because it reminds me of corporate structure.
Oh, how so?
Well, if you are analyzing a massive multinational corporation, you don't track the daily emails of every single employee to figure out the company's overall strategy.
No, you drown in data.
Right. You look at whole departments, you observe how a budget cut in the marketing department directly correlates with a hiring surge in the accounting department.
That is the exact same principle. DSPN's coarse graining creates a top-down modular map of cellular strategy. Instead of micromanaging every gene, it watches the departments.
That's fascinating. Does the paper give a good example of this corporate strategy in action?
It does. The researchers highlight a specific scenario where a cell experiences damage to its ribosomal subunits.
Okay, so a crisis hits the factory floor. What does the coarse-grained map reveal in that scenario?
It shows an immediate coordinated shift in resources. Without any prior manual annotation by the researchers, DSPN's module-level map shows the cell explicitly down-regulating its translation module.
Meaning it stops creating new proteins.
Exactly, it slashes that budget.
Yeah.
And simultaneously, it upregulates its degradation module to clean up the mess. It captures the high-level strategic logic of the cell responding to an internal crisis to maintain homeostasis.
So we have these elegant shortcuts, pseudo-likelihood bypassing the computational bottlenecks, and coarse graining structuring the data into readable corporate departments. But, every time you approximate and group data, you risk losing vital fidelity.
That is always the worry in computational biology.
Right. So the real test is the scoreboard. Does this top-down map actually survive contact with raw benchmarking data when tested against the older algorithms we mentioned earlier?
The performance data in the paper is honestly staggering. It is definitive.
Yeah.
The researchers tested DSPN against PIDC, GRN-Boost2, and GNA3.
The old pairwise tools.
Yeah. When tasked with predicting the top 10 most critical regulatory edges in a given network, DSPN achieved an average accuracy of 96%.
96%.
And the older algorithms.
They hovered between 77 and 83% accuracy on the exact same data.
That is a massive leap.
It is. And the performance gap widens even further as the network scale increases. On a complex 250-node synthetic modular network test, DSPIN scored an accuracy of 0.944.
Okay, let me guess. The older tools completely fell apart.
Pretty much. The highest scoring alternative tool managed only a 0.553. Wow. A jump from 55% accuracy to 94 isn't just an incremental software update. That completely validates the decision to abandon the old pairwise approach.
Absolutely.
It proves that adapting the statistical physics model to map the energy landscape successfully circumvents that masking effect we talked about.
It definitely does. But the accuracy of the structural mapping, while incredibly impressive, only sets the stage for the true breakthrough of this framework. There's more. Oh, yeah. If we connect this to the bigger picture, the holy grail here isn't just drawing a map, because DSP-N models the underlying probability distribution of the transcriptional states. It actually operates as a generative model.
A generative model,
Okay.
meaning it moves beyond just analyzing historical sequencing data to actually synthesizing new data.
Yes. The older tools produce static maps. They just output a rigid graph indicating that gene A influences gene B, end of story.
Right.
DSP-N, however, provides a working computational simulacrum of the cell's decision-making engine.
Okay, so because it has mapped the full topography of the cell's energy landscape, the hills and valleys, you can introduce new theoretical perturbations into the model.
Exactly. And you could simulate how the cell will roll down that landscape into a new state.
So wait, what does this mean for you and me? To put that in concrete terms, if I have physical data on how a cell reacts to drug X and physical data on how it reacts to drug Y,
Right.
can DSPI simulate the cellular response to a combination of drug X and drug Y, even if that specific combination has never actually been mixed in a physical laboratory?
That is exactly what the framework is designed to do. And the paper demonstrates DSP-N's ability to infer the network structure of a cell population and subsequently predict the single-cell transcriptomic output of entirely unobserved combinatorial perturbations.
That is mind-blowing.
It utilizes sampling methods akin to Langevin dynamics to generate completely new transcriptomic profiles based on the mathematical rules it learned from the initial data.
So it generates a simulated distribution of individual cells, predicting exactly how their gene expression will shift under the stress of a novel drug combination.
Yes.
We are basically looking at a highly advanced flight simulator, but for human biology.
That's a great analogy.
You can program 1 million virtual flights or virtual drug interactions and observe how the cellular systems hold up under distinct pressures without ever needing to touch a physical petri dish. So are we looking at the end of traditional petri dish drug testing?
The implications for fields like oncology are massive. Currently, discovering effective combination therapies requires physically screening millions of compounds across physical cell lines.
Which is incredibly expensive.
It is an arduous, resource-intensive process.
Yeah.
A generative model like DSP-N allows researchers to perform high-throughput virtual screening. You can ask the system to predict the efficacy of complex multi-drug cocktails at varying dosages across specific genetic profiles, and you get answers in a matter of hours.
And crucially, because DSP-9 actually maps the mechanistic logic of the network, it isn't just a black box spitting out an efficacy score, right?
It's not just a blind guess.
It provides mechanistic interpretability. It shows you the specific departmental shifts, like the upregulation of the degradation module we mentioned, or the suppression of a signaling pathway that explain exactly why the simulated drug combination works.
It bridges the gap between predictive power and biological understanding. You get the simulated outcome, and you get the detailed roadmap of how the cell arrived at that outcome.
Wow. Okay, let's take a moment to recap the ground we've covered, because this is dense, fascinating stuff.
It really is.
We started with the foundational problem. Mapping gene regulatory networks using traditional pairwise algorithms is like trying to trace a city's traffic routes by indiscriminately dropping barricades and blocking intersections.
Right, it's just noisy.
Very noisy. The structural noise, specifically the masking effect, obscures critical activating interactions because the perturbations shatter the cell's modular coordination. So to solve this, the researchers behind DSP-N borrowed from statistical physics. They applied Markov random fields and the principles of associative memory to map the cell's continuous energy landscape.
And by utilizing pseudo-likelihood to manage the massive computational load, and ONMF to coarse-grain the data into clear, functional corporate departments, DSP-N cuts right through the noise.
And the results speak for themselves.
They do. It delivers unparalleled accuracy in structural mapping, and, most importantly, it operates as a generative model capable of simulating unobserved cellular states.
It is a framework that fundamentally shifts our entire approach to systems biology. And for anyone tracking the trajectory of healthcare, frameworks like DSAPIN represent the foundational architecture required for truly personalized medicine.
Oh, absolutely.
I mean, in the future, sequencing a patient's tumor could allow oncologists to build a bespoke DSPN model. They could run thousands of virtual chemotherapy simulations to find the precise combinational therapy that collapses that specific patient's network.
It transitions medicine from a reactive discipline, heavily reliant on generalized clinical trials, to a predictive science tailored to the individual's unique biological topology.
Which leaves a final, rather profound question to consider for everyone listening. We began by comparing this process to drawing a map of a city grid. But as generative models like DSPI grow more sophisticated, expanding from single cells to entire tissue systems, the resolution of that map will just continue to sharpen.
That's the trajectory we are on.
Right. So if we eventually reach a threshold where a digital generative simulation of your cellular network can predict your response to any disease, pathogen, or pharmacological intervention with near-perfect accuracy, at what point does your digital biological twin become a more functionally accurate representation of your health than the physical body you are currently walking around in?
That is wild to think about.
Something to ponder next time you pop a perfectly standardized, mass-produced painkiller. Thanks for joining us on this deep dive. Until next time.